Let us do Brahmanan.
Hello students, in the previous session we discussed the types of acquired immunity, that is on the basis of place of production of antibodies. Acquired immunity can be classified into active and passive immunity on the basis of place of antibody production. In active acquired immunity, the antibodies are produced in own body, whereas in passive immunity, the antibodies produced in the body of other animal or human is administered for a specific infection or disease. Active immunity again natural or artificial. Natural active immunity is developed to an infection. For example, the polio subsequent infection that leads to the poliomyelitis. Similarly, the artificial active immunity is developed in response to a vaccine. For example, the resistance to tuberculosis developed by the administration of BCG, like BCG. And the passive immunity, again of natural or artificial. The natural passive immunity is like the transfer of maternal antibodies to the fetus before birth or to the infant or newborn baby after birth. Before birth, the immunoglobulin G can pass through the placental barrier. IgA that is present in the colostrum, in the breast milk, that provides immunity because the immune system is not developed in the infant, in the early infant. We discuss the lymphoid organs, which are two types, primary lymphoid organ and secondary lymphoid organ. Primary lymphoid organs are bone marrow and thymus gland. And the secondary lymphoid organs are the lymph nodes, the tonsils, the MALT, M-A-L-T, mucous-associated lymphoid tissue, spleen. Spleen is the largest lymphoid organ. These lymphoid organs, the lymphatic tissue, the lymphocyte, they make the immune system. The immune system disorder is characterized by allergy. What is allergy? Allergy is the acquired abnormal hypersensitivity or hyperimmune response to a foreign agent, which is called allergen. The different allergies are known to be hay fever, asthma, eczema, and anaphylaxis. Whereas the allergens are like the dust particles, the pollen grains, the chemicals, cosmetics, even the food like yolk and the milk. Means not to every person. Some person are allergic to some substances, foreign substance. We discussed the immune system disorder is due to the overactivity or low activity. Overactivity is known as hyperimmune response that causes the autoimmune disorders like myasthenia gravis, multiple sclerosis, rheumatoid arthritis, or the low activity leads to deficiency disorder, which may be again that primary and secondary. Primary is due to the genetic cause. If there is a mutation or any genetic disorder like XLA, X-linked A-gamma globulinemia, which led to the non-maturation of B and T lymphocytes and leading to the destruction of the specific the immune system of the body. And the secondary is secondary deficiency, immunodeficiency disorders are like the due to the malnutrition or infection, like cancer, example AIDS, etc. today's session we are going to discuss vaccination and immunization. What is vaccine and types of vaccine? Okay, what is vaccination? Vaccination is the administration of a vaccine to help the immune system for developing the protection to a disease. Vaccination vaccination it is the administration of a vaccine to help the immune system for developing protection against protection or resistance against a disease. What is administration? Is the introduction of the vaccine into the body. It is the most effective method. It is the most effective method for prevention. Most effective method of prevention of infectious diseases. It is known that the worldwide eradication of the smallpox, worldwide eradication. Do you know eradication? Eradication is the complete destruction. Eradication of smallpox or and the elimination of polio and tetanus. Polio, tetanus, which are the infectious diseases, is possible due to vaccination. So, vaccination is the act of introducing vaccine into the body. We can say more precisely, we can define vaccination is the act of introducing a vaccine to stimulate a person's immune system to provide the active acquired immunity against a infectious disease. Okay, so vaccination is the act of introducing a vaccine into the body to stimulate one's immune system to provide active acquired immunity against a specific infection or disease. Then what is vaccine? Vaccine is what is vaccine then? Vaccine is the biological preparation of the pathogen. Vaccine, a biological preparation of attenuated or inactivated preparation of attenuated or inactivated pathogens like bacteria and virus, or pathogen like bacteria or virus or its toxin or the isolated or an isolated antigen. Isolated antigen, they are the surface proteins. Proteins present on the plasma membrane of the pathogen. Surface protein. That stimulate stimulate the development of stimulate the immune system to provide active acquired immunity against a disease, against a specific disease, against a specific disease. We know what is vaccination, what is vaccine. Vaccine is the biological preparation of the pathogen, attenuated or inactivated pathogen, whereas vaccination is the administration of the vaccine to a body to stimulate the immune system to provide the active acquired immunity. Now, what is immunization? What is immunization? It is a process by which a person is protected from protected from a disease, protected from a disease through vaccination. However, you see, the principle of vaccination and immunization, the principle of vaccination and immunization is based on a property of memory of immune system that works on memory basis. Okay, the principle of vaccination and immunization is based on a property of the memory of the immune system. Let us see how the vaccination how it is done. The attenuated or inactivated form of pathogens are administered, then body gets immunized, immunization is done, then after immunization the body produces either antibodies against that specific pathogen that contains the vaccine, or the immune system that develops the body produces the lymphocytes, memory cells that develops the memory cells, that is memory B cell or memory T cell. Then they produce on the subsequent exposure of the body to that pathogen, the memory B cell produce the antibodies. On the subsequent exposure, then the memory cells produce antibodies because the memory cells recognize exposure. Memory cells recognize the pathogen. They recognize the pathogen. That's why they produce antigen. Okay. So, how the pathogens are attenuated? And we discuss all this. The vaccine term is derived from where the vaccine term is a vaccine, then vaccination is derived. Vaccine term is derived from a Latin word vacca, means cow. Edward Jenner coined the term vaccine. For the first time, Edward Jenner has observed that if the persons are infected by the cowpox, like he observed the the milkmaids, they were not they were not contracting the smallpox if they were already contracted the cowpox. Cowpox generally infect the cattle. Okay, so Edward Jenner first he found that cowpox, if the persons are infected by cowpox, they are not again infected, they contract the smallpox. So he he immunized a boy with the cowpox virus collecting from the blister of the cow, and he found that the boy then after that he did not get the smallpox throughout his life. So Edward Jenner also coined the term vaccine. He developed he first immunized, he prepared the vaccine, first done the immunization, hence he respected as the father of immunology. Edward Jenner is known as father of immunology. Immunology, study of immune system and its function, how the immune system function, this vaccination, immunization, all this coming under immunology. But actually the technique of attenuation of pathogen was first developed by Louis Pasteur. Okay, so we came to know that the vaccination immunization, they work on a memory basis. Okay, the immune system work on a memory base. The types of vaccines: natural live vaccine, live attenuated vaccine, inactivated vaccine, toxoid cellular fractions, the antigen live vector vaccines, the recombinant antigen vaccines, DNA vaccines, polysaccharide vaccines, combined vaccines. Let us discuss one after another. Natural live vaccines. The example are cowpox virus vaccine, simian bovine retrovirus vaccines. Cowpox virus vaccine already we have discussed. The live, see, no alteration, no modification. The natural live the non-pathogenic natural pathogens, non-pathogenic condition. Okay, they are taken live to make the vaccine. Simian retrovirus vaccines are prepared from simian virus. They generally cause to monkeys. They infect the monkeys. Bovine simian virus generally causes a fatal immunodeficiency in monkeys. Bovine in cattle. They cause this simian retrovirus, bovine retrovirus, they cause a fatal immunodeficiency disorder. Fatal immunodeficiency lead to death. Okay, the bovine virus in cattle and simian retrovirus in this bovine retrovirus is equivalent to HIV of human. Okay, the live attenuated vaccines, the live attenuation of the pathogens, how it is done? It may be done, the attenuation is done through chemically or physically. Chemically by using formaldehyde. By using the formaldehyde, the pathogens are attenuated, made less virulent. Physically how it is? Through heat. By using the heat. By heat treatment. Chemically means keeping the pathogens in formaldehyde. Okay, washing the pathogens in formaldehyde. Physically means through heat treatment. And the examples are BCG, already we have discussed, Bacillus calmette-guerin or Sabin polio vaccine virus, that is Sabin poliomyelitis vaccine for yellow fever and measles. These are the vaccines are prepared from attenuated or that weakened pathogens. And inactivated or killed vaccines, example is Salk polio vaccine or vaccine for pertussis or whooping cough. Toxoids, the toxoids are taken from the diphtheria and tetanus bacteria. Diphtheria and tetanus. Diphtheria is pertussis is the whooping cough. Toxoid. Diphtheria and bacteria toxins, they develop the passive immunity. Generally, as we have discussed, ATS, anti-tetanus serum, anti-diphtheria serum, these are this immunity, the vaccines are used for passive immunity. Used for passive immunity. The cellular fractions, meningococcal vaccine or MAN-B, These are developed from the polysaccharide extracted from, you see, polysaccharide. Vaccine is prepared from polysaccharide of the cell wall of the meningococci. Meningococci bacteria. Okay, from there the polysaccharides are extracted. Cellular fractions. These polysaccharides make the cellular fragments of the cells. They are taken for the preparation of the vaccine. Okay, now the combined vaccine. Remember one thing, this meningococcal vaccine is given to all kids. Given to all kids. Recently developed, but very much efficient and safety. Efficient and safety vaccine. Okay. Now let us discuss the combined other vaccines. Like the combined vaccines can immunize for more than one infection, or the vaccine containing more than one immunogenic agents. More than one immunogenic agents. Here, DPT, diphtheria, pertussis, tetanus, diphtheria, pertussis plus tetanus. So, the three type of immunogenic agents are there. Three immunogenic agents. Here, two immunogenic. What are they? Diphtheria plus tetanus. MMR, measles, mumps and rubella. Measles, mumps, rubella viral preparation is done. That is 3-in-1. Polysaccharide vaccines. These are prepared from the vaccines prepared from capsular polysaccharide. For example, Haemophilus influenzae, Streptococcus pneumoniae. These are excellent antigenic substances. They make the excellent vaccine. Excellent vaccine. Good immunogenic. They create the good immune response. That's why called excellent vaccine, that create the good immunogenic response, act as good antigen. Or good antigen that creates excellent immune response. Okay. live vector vaccines. How they are produced? The antigenic, the gene coding for antigenic protein introduced into the vector, joined to a vector. What type of vector? The vector may be the attenuated bacteria or virus. The vector may be attenuated bacteria or viruses. The gene is joined to a vector or introduced joined to a vector, then they are inoculated. These are transformed. The new that the gene is joined with the vector, then it is transformed, called transformed vector. They are inoculated or introduced. Inoculated or introduced into an individual. Within it, the vector replicates. Vector replicates, and vector slowly replicates and serve as a source of antigen. Vectors replicate slowly and make the source of antigen in the individual. Serves as a source of antigen in the individual, and so the immune response is developed. In recombinant antigen vaccine, how it is, the recombinant type is formed? Here the gene coding for antigen protein introduced into the yeast or bacterium or mammalian cell. Gene coding for antigenic protein or antigen protein, okay. Introduce into yeast, bacteria or mammalian cells. Then that recombined with the genetic material of the yeast, okay, yeast of these yeast or bacteria or mammalian cell, and within it and how it is introduced by using the rDNA technology. Introduced and by rDNA recombinant DNA technology, by using rDNA technology. Then what happens? The antigenic proteins are produced. These cells, yeast or bacterial mammalian cells, are cultured in laboratory. And, What happens? Listen. Gene coding for antigenic protein are introduced into the yeast bacterial mammalian cell, and they are incorporated through the rDNA technology. They are introduced. Genes are introduced. Then these cells are cultured in laboratory, and there these genes express the proteins, and proteins are synthesized. Antigenic proteins are synthesized in each cell, either in the mammalian cell or in the yeast cell or the bacterial cell. Then they are harvested to prepare the vaccine. Then vaccine is prepared. Okay. This recombinant antigen vaccine is developed by using the rDNA technology. Here you remember the surface antigens are taken. Surface antigens are taken, are produced or developed. Gene for surface antigen is taken. Now let us discuss the DNA vaccine. The gene encoding the desired protein is taken. So we can say desired DNA vaccine. Let us see, this is the recent type vaccine. The desired gene is extracted from the pathogenic agent and plus the plasmid. Now, recombinant plasmid, that is introduced in the muscle cell. Within the muscle cell, it start expressing the antigen. Within the muscle cells, expression of antigen occurs. Expression of antigen within the cell. Then that develops the antigen directly. The antigens, they are released into the tissue and that cause the development of immune response, that cause the development of immune response in a natural way. Of course, it is artificial, but the gene for that antigen, the desired gene which expresses the antigen, is incorporated with the plasmid through the rDNA technology. Recombinant plasmid is produced, that is introduced into the muscle cell. Within the muscle cell, the antigen are produced. Directly the antigens cause the development of immune response in the body. This is how we discuss the different types of the vaccines. We discuss the vaccination, that is administration of the vaccine. Immunization, the process by which the body gets immunized, protected from a disease through the vaccination. And mainly the vaccines are developed from attenuated or inactivated, that means weakened or killed pathogens or from the toxins produced by them. Okay, thank you.